All right, everyone, I'm really excited to introduce Dr. Heike Rebholz, and she's going to be talking about some mouse models of OCNDS. So please welcome her with a round of applause.
Hello, nice to see some of you again. Nice to see new people. Yes, I'm Heike. This is my third OCNDS conference, and it gets more exciting and more research, more science every time. It's a really great time, I think, now for OCNDS. So I want to talk today about our work that we're doing, and we're focusing on mouse models of the condition. That means we're trying to understand from mouse lines that we generate how, if they represent the patient symptoms, and how what we can understand from these models in terms of how the disease is actually mediated. What are the mechanisms that cause the disease to be expressed or the condition to be expressed? So couple of sections. First, an introduction on the mouse lines that we have and why do we have these mouse lines and not others. And then I will give you the data on the first mouse line that we have characterized and which has worked pretty well. And yeah, is a good model, we believe, to study the condition. And then we have recruited more mouse models to compare whether they have similar symptoms, what's the differences between them. And in the second part, I'm going to talk about a study that have basically spun off of the characterization of the first mouse model, which is understanding gastrointestinal symptoms in the mice. And that's quite an exciting project right now that we have in the lab. Okay, so CK2, do you want a little bit of an introduction? I guess you don't need one because there were several talks before me, but it's a kinase that's ubiquitously expressed. That means it's in every cell of our body. That's also highly active and has a lot of substrate. That means it's interacting with a lot of proteins in our cells and phosphorylates them, adds a phosphate group to these proteins. So there is we know of a multitude of substrates that get that job done, that get phosphorylated by CK2. And that makes it also hard to understand which ones exactly are those ones that are causing the condition to be expressed. And that could actually be potentially used for targeted approaches to help OCNDS patients or for therapeutic approaches. And what else? We already know that it's linked to other neurodevelopmental disorders such as autism. There have been publications about CK2 phosphorylating proteins that have a role in neurodevelopment already. But we also know it's important, for example, in inflammation and in metabolism of cells. So here is the scheme of the protein CK2 alpha, so the one that's encoded by CSNK2A1. And it simply should show you that there are different point mutations, and you all are, families with patients, so you know that there is not only one gene variant that causes CK2 OCNDS, but there are a multitude. But what's interesting is, sorry, I'm going to take the what's interesting is that they're not evenly distributed, but we have hotspots of variants, of mutations. And the number one hotspot that approximately 30% of patients exhibit is that 198R mutation. So it was clear if we want to study OCNDS, we have to start with this mouse line because it's representing the majority of patients. But we also, in terms of better understanding of the functioning of the protein, we decided to look at a mouse that has a mutation knocked in at the N-terminal domain of the protein, which is important for basically interaction and for its function as a kinase. We've also decided we're going with another mutation that's a hotspot mutation, so it's present in a multitude of patients at the other end of the protein, at the called C-terminal end, because we know that mutation, for example, leads to protein instability. So these are different mutations from which we have already, just by their location, a little bit of an insight of what might be not functioning 100% well when they're mutated. And so we decided to go ahead and generate 3 mouse lines, and one was already made. So the 198R mutation that's present in 30% of patients, then the N-terminal one, the 47G, and the end one, the C-terminal one, which is called the 312W. And then we also had a knockout mouse line that had already been generated by colleagues in Boston many years ago, that we also wanted to study. Knockout means this mouse only expresses one gene on one chromosome for CK2, and therefore the protein that's coming out at the end is less present because we only have one DNA strand from which that protein can be generated. So a knockout mouse in our case is we have less active CK2 protein there, approximately 50% to be expected less. So the first mouse line that represents these 30% of patients, you can already see with our bare eyes, we see which one is the mutant, which is not the mutant mice. So the OCNDS model mice, they are a little bit smaller, and as they grow older and get fatter, they're also thinner. So we can see when the mice are old just by looking at them and comparing them, which one is the mutant mouse, so the OCNDS mouse, which is the control mouse that's supposedly healthy. So these are the growth curves. And you see that the lower part means the heterozygous or the mutant mice are smaller. We see that for males and females. And these are photos of embryos that we have taken out of the pregnant mom. And what we saw doing that some of those OCNDS model mice, they're even not being born. So heterozygous mice means they are carrying that mutation just like a patient on one strand of the DNA. And that's what we're also studying. These are our model mice. And so we expected them to be born at a normal ratio as expected. However, we have some prenatal loss, which indicates, that there are some periods during development where it's very important to really have just the right amount. And maybe those heterozygous mice that are absorbed during pregnancy, there's something a little bit below a threshold that doesn't make them get born. But once they're born, they're doing pretty well. It's just they're a bit smaller, but they're doing, they're fertile, they're growing, they have a lifespan that's tiny bit shorter, but not really at all significantly shorter. So we looked at the brain because we want to study the brain of those mice, and overall there was no significant difference. The brain structure was the same. The layers within the cortex, within the outer regions, the outer layers of the brain were similar. We couldn't find a difference except one, which is that the ventricle, so that's where the cerebrospinal fluid is accumulating, that was a little bit bigger in the mutant mice, so the OCNDS mice, compared to wild type. We have not really followed up on that. But so in general, the brain structure, cross structure that we can see and measure is not altered. Then we did some behavioral testing because in the end, what we want to have those mice for is we want to have what we call phenotypes that reproduce the patient symptoms somehow, that we can then go in later if drugs are developed and say we give the drugs to the mice, can they actually help? Do those behavioral phenotypes, can they be rescued? And so we first need to establish which of those phenotypes or symptoms can we detect in the mice. And so that one is a memory test where basically we induce a fear state into the mice by giving them a tiny electric shock. And then the next day, we put the mice back in that environment and we just measure the amount of stress that the mouse has by freezing. So if the mouse remembers well the stressor from the previous day, it will sit there and be stressed and freeze. And there we saw actually that the mutant mice, so the OCNDS mice, don't remember that well. So they have a memory deficit, or I'm not sure if that translates to human at all, but it's for us an important phenotype that we can later study, for example, if we get into drug studies. Other phenotypes are that they're more hyperactive, the mice. that they, when they're asked or when they're put with a with a cotton piece of cotton, they're naturally building a nest, and they're less able to build a nice, beautiful nest than the wild type littermates. But they're still building a nest. But it's when we score the quality of the nest, it's just not quite as good. And then that is very interesting is because we have different mouse models of OCNDS, and one symptom or phenotype that we reproducibly see is when we induce them into a seizure state, into an epileptic state, by giving a drug. They do that, the heterozygous, so the OCNDS mice do go faster into seizures, and they have stronger, longer seizures, and sometimes also more lethal seizures. But don't worry, it's just the mouse status. Often when they go into a seizure, they can't handle that. So there is a phenotype with seizures that, of course, 25% of OCNDS patients have too. So just summarizing, these main phenotypes are, well, and we're not at all done. This is the first mouse line, and we're still working much more on the level below the behavior. We're doing biochemistry. We're, trying to get at mechanisms. We're analyzing the proteome by phosphomass spectrometry, etc. But what we can say so far, that for this mouse line, we can somehow say, yes, we can detect a developmental delay. We have done cellular studies that show that the neurons mature less fast, derived from the OCNDS model mice. We know that they have some cognitive defects. They have some stereotypies. These are uncontrolled movements that are not goal-directed, that we can detect those. We don't detect motor and sociability phenotypes. So that's something I'm not sure if you want to, we can discuss that later, but we don't detect, for example, hypotonia, which seems a really common phenotype or symptom in the patients, but we don't detect that in the mice. But that said, we didn't try it with newborns. We tried with very young adults, so that might also be something where we missed the window of testing. But so far, these two groups of experiments haven't shown anything relevant for the disease. We have found the seizure phenotype very clear. And for the sleep comorbidity, we have seen slight changes when we record the mice over several days. We have seen activity changes, but we haven't seen changes where we say they really sleep at different times or they sleep less, but they're more active when they should be actually sleeping. That we have seen. So to understand, the aim is for us not only to reproduce the symptoms so that we can test drugs, but also, of course, to understand how it comes about that the disease, the condition is expressed. So what are the molecular underpinnings, right? And so one way to get at that in an unbiased manner was to study the phosphorylation status. So kinase is doing that job of adding phosphor groups to a whole bunch of proteins. So we took all the proteins from a brain region, and then we analyzed that for how the phosphorylation is present on all the different proteins that are in the cell. And then we run these data. Of course, we say, okay, there's a difference between the model mice and the wild type mice. And we run these differences in an algorithm to actually categorize to which functional groups do those proteins that are changed belong. And very clearly, the top candidate that we see in not only the 198R mouse line, which is shown here in red, but also the three additional mouse lines that I introduced in the beginning, they all converge onto this synaptic compartment in the neuron. That means we're expecting that at least part of how the disease manifests itself is through problems at the synapse. And the synapse, you probably have heard of what a synapse is, but it's basically the point of communication between two neurons, whereby one neuron receives an input through neurotransmitters that are released from the incoming neuron. that has a presynaptic terminal where it releases chemicals that are then recognized on the postsynaptic side by the receiving neuron. And then these neurochemicals they're called neurotransmitters, they lead to changes starting from expression of other proteins to actually propagating a signal into other brain regions. There are many things, but this point here, that point of communication, that synapse, seems to come up in the 4 different mouse lines as most affected. So that's interesting because it tells us, yeah, we have 4 different mouse lines, but we're looking at possibly something quite similar. So this is where we're going right now in our studies. So part 2 is this comorbidity that a lot of your children probably experience, which are gastrointestinal issues. This is just a slide to show you that there is always core symptoms, and then we have, this is actually taken from an autism talk of somebody else. I took it from the literature, but it shows you that there are, core symptoms, and then we have a lot of comorbidities that come along, and some can be very disturbing and making your life difficult. So we actually were interested, well, based on data that was published, and based also on a survey that the foundation put out about how many of your patients, your children, suffer from GI problems. The number was quite high. So we were interested to see in our mouse model what happens to the gut, right? So the gut of the mouse is more or less the same. It has intestine, colon, etc. Looks a little bit different because we have the cecum here, but we could study we were in the process of studying the gut, and we, for example, could see that there is more food pellets in the colon of the mice, not food pellets, fecal pellets in the colon of the OCNDS model mice compared to wild type. Could that indicate constipation? Well, possibly. We don't know, but that's just the first observation. We then cut slices of the colon and looked at that after staining with different dyes. And we saw a couple of things. We saw that the muscle layers are changed in their thickness, which again could stand for that the muscle is just not working as well to expel fecal products. So that is an interesting finding. We believe, and maybe possibly more interesting even, is that the number of, they're called goblet cells. These are cells in the colon, there's these invaginations, and within these invaginations, here we have crypts, and in these crypts there are goblet cells, and they're important to release mucosa, and the mucosa is super important to protect the lining of the gut from bacteria, from toxins, from whatever, and also just having the passage of fecal products enable that, right? So we found less goblet cells, which would indicate that the mucosa is not as intact, less, and that possibly there could be an inflammation because the bacteria that, of course, live in our gut, they make up the microbiome. They actually, shouldn't really talk to our blood system. They should be just in the gut and not have access to the blood system. So this is what we found there. You have probably, the microbiome is a big sexy word in many papers, even fashion magazines, whatever. You find the gut microbiome everywhere, and it's actually true that it is quite an important entity in our bodies that can mediate behaviors that are caused by the brain, right? So, for example, it all started when people worked with mice that have never been exposed to bacteria. They are called germ-free mice, and they had behaviors that were abnormal. So we started to understand that the gut and the brain, they communicate, and that's actually true and now very well established, that the gut microbiome, when it's not in balance, can have effects, detrimental effects on the brain. It can cause inflammation in the brain. And there are 2 ways of how gut and brain talk to each other. One is a nervous system effect that's called the vagus nerve. And the other one simply through that endothelial lining that lets metabolites pass into the fluid, the interstitial fluid, and finally ending up in the blood. And then the blood can talk to the brain as well via the blood-brain barrier, can exchange molecules and macromolecules. So there are two ways. So we started out first to ask, do the mouse model, yes, I know I'm almost done in 34 seconds. Do our model mice, do they have changes in the gut microbiome? And actually they do. We find, for example, 2 strains reduced in their strength or in the numbers of bacteria that produce short-chain fatty acids that are important, actually known for gastrointestinal health. So yes, we have a difference in the gut microbiome. So, and I'll skip that. And we have indications from there that we have inflammation that's systemic, that's in our blood. We have, for example, enlarged spleens, which are the organ in us that filters the blood, but also produces white blood cells. So we have a strong indication that there is inflammation. There are inflammation processes going on. We see that also by just checking for inflammatory molecules in the blood. So we know OCNDS model mice have got morphological, histological differences, that they have an altered microbiome, and that they have inflammation. So what we want to do, and what is very important to really make our story or our work worthwhile, is to really say that's reproducing what we see in patients. That's also happening possibly and underlying the gastrointestinal symptoms that the patients experience. And for that, we have planned, long time ago, started to plan that human study where we collect actually fecal tissue, basically feces from children affected and a control person from the family. And this is actually happening today and tomorrow at this conference. And really, it makes a huge difference to step out of the mouse work into human work. It makes it much more relevant, and that's why I almost beg you or hope that you are trying to take part. I know it's not so easy, but if it's possible, we would all benefit from that. So with that, I basically thank you, and I want to also thank, of course, my team. We're based in Paris. Thank you.